wszystkich na czwartym dniu tygodnia mózgu. Welcome to the fourth day of our Brain Awareness Week. So today I have a great honor to introduce our first keynote speaker, Professor Zoltán Molnár from University of Oxford, one of the most outstanding and globally recognized scientists working in the field of developmental neuroscience, mainly the cortical development, neocortical development, and interactions, neocortex, thalamus, and so on. Professor Zoltán Molnár is professor of developmental neuroscience at the University of Oxford and fellow at St. John's College. He's known for key contributions to our understanding of how the birth of cortical neurons is regulated, how they migrate, differentiate, generate axons, and assemble into circuits, and how those pathways change over time, partly as a result of activity passing through them. Professor Molnár earned his MD at the Albert Szent-Györgyi Medical University, Szeged, Hungary, and Doctor of Philosophy at the University of Oxford. He also investigated thalamocortical development working at the Institut de Biologie Cellulaire et Morphologie, Université de Lausanne, Switzerland, and learned optical recording techniques to understand early functional thalamocortical interactions at Kyoto Prefectural School of Medicine in Japan. He was awarded the title Professor of Developmental Neuroscience in 2007. Professor has been elected member of Academia Europaea in Physiology and Neuroscience, European Neonatal Brain Club, Fellow of the Anatomical Society, awarded New Fellow of the Year Award for 2018. So please welcome Professor Zoltán Molnár.
Thank you very much, Arthur, for your very kind words, and also I would like to say thanks to Kasia for looking after me yesterday. And yesterday, Kasia's daughter said that she didn't understand too much of Kasia's lecture, so I've redone my lecture, so I will do quite a bit of introduction before I home into the shadows of the subplate, which is my main research area today. So I don't know whether you know what is the origin of the word neurology. Where is this word coming from? It's coming from Oxford, of course. That's why and if you visit Oxford, which I hope you will do, and if you walk on this street, it's called the Merton Street, and it didn't change, the street probably didn't change over the years. This is Merton College, chapel, and this is one of the oldest colleges, 1260 or something like that, when it was established. And opposite the lodge, you have two houses. One of them is the Postmaster Hall, where William Harvey worked, and next to it there is another house, Beam Hall. This is where Thomas Willis lived and worked. So Thomas Willis is my hero. He is founder of clinical neurology, neuroanatomy, comparative work. Working in this house, he wrote his book Cerebri Anatomy in 1664, and this is the first mention of the word neurologia. And then later it was translated by Partridge in 1681 as neurology, but neurologia, the word, appears in this book first. So have a look how Willis was working in this place, and these are his friends. Christopher Wren, the architect, who was the architect behind St. Paul's Cathedral, lots of colleges, All Souls College in Oxford, for instance, and he did the illustrations for Willis. Then Lower, he was the student. He did the first experiment when they ligated the vagus nerve in a dog, and the dog died. But they noticed that it had an effect on the heart rate. And Robert Boyle, the chemist, the Boyle law, can you imagine the density of people living in Oxford and working together to understand the brain? So that's why Willis is so well known. And now you know that the word neurology is coming from his book. Every medical student has to learn the name Willis because of the circle, the arteriosus circle. But he was not the first. He was in fact number 5 who described this structure. But he had the best illustrations because of Wren. So Christopher Wren was making these drawings. But the reason why I usually start my talks with Willis is not because of this circle, but because he was the first who suggested that it has to be the cerebral cortex, which is the seat of higher cognitive functions. So Willis was comparing a sheep brain with a human and many other species, and noticed that although the brainstem was very similar, the cerebral cortex was much bigger and more convoluted. And he suggested that the cerebrum is the primary seat of rational soul in man and the sensitive soul in animals. It is the source of movements and ideas. And he got it absolutely right. Our cortex is basically the seat of our higher cognitive functions. And today I'm going to talk about the development of this structure after introducing this topic a bit more. So as you know, the human brain is about 1,350 grams on average. It has millions of cells with billions of connections with trillions of specificity. And if you cut a brain open and then you have a look at the structure, you also notice that about 80% of the volume of the brain is the cerebral cortex. So and on the telencephalic vesicles. And if we zoom through the human brain in the serial MRI images, you can see just how big the cerebral cortex is. And it's responsible for, as I mentioned, 80% of the volume of the brain. And no wonder that Willis realized that it is big and convoluted. But the human cerebral cortex is indeed very big and convoluted, but it's not the biggest and not the most convoluted brains. There are other brains which are bigger than the human brain. So what is so special about the human brain? And it wasn't until later when the techniques developed to stain cells in the brain, like Nissl staining, that they understood that the cell numbers are very special in primate, and also it's very special for the human. And Arthur just showed me the picture of Susanna Herculano-Husel, who was visiting also, and it was Herculano-Husel and Roberto Lent who developed methods to count the cells in our brain. So they realized that the cell density and cell numbers change across the brain, so it's very difficult to count the cells just by looking at one region. So what they did was they made a soup out of the brain, so they digested the brain and they made an isotropic soup of this heterogeneous structure. And then if you take a small volume of this and you put it in a Burkert chamber, then you can actually count the cells. And if you use a general nuclear stain together with maybe a transcription factor which is expressed in neurons like nu-n, then you can actually count the cells. So you make it isotropic, and then you, with a known volume, you can then calibrate the cell numbers. And from this work we know that although the cerebral cortex is 80% of the volume of the brain, the cell numbers are basically just maybe 20%. So where are all the cells? And it turns out that it's actually in the cerebellum. The cerebellum has, it's only about 10% of the volume of the brain, but 80% of the neurons are here, so very small, large density cells are in the cerebellum. But nevertheless, the primate cerebral cortex and brain has the largest number of neurons. So if you do the same exercise in an elephant, they have a much bigger brain, but they have half the number of neurons because they are bigger and also loosely packed. Whereas our cells have a different scaling rule. Primates, we have small neurons and very densely packed. And we are just a large primate, basically. a very similar density in a chimpanzee, but our brain is a bit bigger. And this is becoming apparent when you compare a rodent with a primate. So an owl monkey and an agouti, it's a South American rodent. If you look at the cell numbers, then we completely outperform the rodents. They have a lower cell density. So humans, we are primates with a large brain, with a high density of cells, and that is making us very different from everybody else. Our brain is evolved that, according to a scaling rule, that we have the largest number of neurons on the planet. So we have the largest number of cells. And so what else is special about the cerebral cortex? So it took quite a few centuries to understand what is the function of the cerebral cortex. And we have to go back to Vesalius, who basically started looking at lesions, because there are, even now, today, the techniques we can use to study brain function is lesion, stimulation, and recording. But we use better and better techniques for all this. So Vesalius noticed that the brain controls the spinal cord, and that the spinal cord is controlling the muscles from all sorts of lesion clinical cases. And also Boyle, he already noticed that damage to the one side of the brain is causing a contralateral paralysis. And also he noticed that lesion in the left hemisphere cause loss of speech. So this has been known for quite a while. And then later on, it was Jackson who was a clinician, and he looked at patients where you had a focal epilepsy and looked at the movements associated with the epilepsy, which became more and more generalized. But he documented that where was the lesion and where did the epilepsy start and how was it marching across the surface of the brain. So from the Jacksonian epilepsies, he already drew conclusions on cortical localization. Then it was Fritsch and Hitzig, the first in Berlin, who stimulated anesthetized dogs, especially in the frontal regions, and looked at the movements of the limbs. And this is how they mapped out the localization in the motor cortex. Ferrier in England, he did the same in monkeys. He was stimulating certain regions and then looked at the responses. So all these resulted to the concept that functions are localized in the cerebral cortex. And the German School of Anatomy, again in Berlin, where Brodmann did his thesis, he was looking at Nissl-stained material of brains, and he compared the differences across different regions in macaque and human brains. So for his PhD thesis, he had to do all this analysis. And basically, this is cytoarchitecture. You are looking at comparing the structure of the cerebral cortex across the convexity. And from this work, we know that there are some big changes in cytoarchitecture in the brain. So if you look at a cross-section, you see that the cerebral cortex looks very similar across. But if you zoom into the banks of the calcarine sulcus, then even sometimes with naked eye, you can see a stripe, which is called stria gennari, because the layer 4 cells here, they have a more myelination, and it's much bigger, this cell layer. So you can see with naked eye, the 17-18 border. So, and that's what these numbers come from, actually, Brodmann. Let's show you macaque cortex. In macaque, the occipital pole is flat, unlike human. And if you cut a sagittal section, you can see that the 17-18 boundary is very obvious. So this is the primary visual cortex, and the secondary areas are much, they have a different cytoarchitecture. And that's exactly what Brodmann was doing for his thesis. He was punching cylinders in different parts of the brain, had a look at the proportions of the layers, and then he moved on to another area and compared them. And you can see that he started his work here, next to the central sulcus, and moved in concentric circles out. And now we are stuck with these numbers. These are historic numbers. We still refer to cortical areas based on Brodmann's original numbers. And the reason why this is such a powerful map of the brain, because these proportions, they reflect differences in the cell types of the brain. And these different cell types, different proportions, they indicate different computational functions these circuits have to perform. So for instance, if you want to wiggle your fingers, then you have to have signal from these layer 5 cells to your basal pons and to spinal cord. You project to the thalamus from layer 6, and you have intracortical connectivity from the upper layers. And in the visual cortex, you have huge proportion dedicated to layer 4, the thalamic recipient cell group, whereas in the motor cortex, where you have much bigger layer 5, you have a very thin layer 4. So that's why Brodmann's name is still with us today, and we still use these numbers even today, although these maps are not perfect, but they reflect the different computational functions we perform with these circuits. There are, of course, many other maps, and it's still going on. Allen Brain Institute is doing it based on gene expression, single-cell transcriptomic, but nevertheless, Brodmann maps are still with us today. And we also know that although the cerebral cortex looks very homogeneous, the cell numbers are very, very different in different regions. So if you flatten cerebral cortex and you cut out individual arbitrary columns and you count the cells, and this has been done by John Kaz using Susanna Herculano-Huzel's technique, he noticed that the visual cortex can have double or 2.5 times more cells than maybe a frontal area. So the cerebral cortex is not uniform. It does look uniform if you just look at it superficially, but it has a very different circuitry, very different cell numbers, and basically we specialize for these different functions. So the cerebral cortical areas, they specialize for the different functions during development and during your life. And this concept also emerged very slowly because the phrenology caused a big damage to the idea of cortical localization, and it took a time to re-establish this concept. And Sherrington, who was working in Oxford and Liverpool, contributed a lot to this field. So what they did was they opened the skull, the dura, of anesthetized monkeys, and then they stimulated in different parts, and they mapped out the representation of the body in the motor cortex, and also sometimes they also mapped out the sensory representation. And from this work we know that the representations change, and the size of the representation is dependent on the density of the receptors or the kind of movement you perform in the motor cortex. And Sherrington had a very famous student, Penfield, and Penfield took this further when he returned to the States, and then later on he moved to McGill University in Montreal, and then he started doing the same technique in human. So once you remove the skull and you opened up the dura, you can wake up the patient because there are no more pain receptors on the brain surface itself. So Penfield decided to map out some of these areas in before epilepsy surgery, and then looked at the representations and using the Sherrington technique, mapped out the sensory and motor representations in the brain. So from this we know where are these representations and how they change. And this technique is still used even today before epilepsy surgery for language areas. And I just show you a brief video from George Orgerman, who is doing it for a patient.
A basic language function, like counting, relies on a widespread network of sites.
9.
If he hits one of these essential speech areas, the electric current will stop the patient in her tracks.
14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 61, 62, 63, 64, 65, 66, 67, 68, 69, 70, 71, 72, 73, 74, 75, 76, 77, 78, 79, 80, 81, 82, 83, 84, 85, 86, 87, 88, 89, 90, 91, 92, 93, 94, 95, 96, 97, 98, 99, 100, 101, 102, 103, 104, 105, 106, 107, 108, 109, 110, 111, 112, 113, 114, 115, 116, 117, 118, 119, 120, 121, 122, 123, 124, 125, 126, 127, 128, off.
17,
Okay.
18,
So number 10.
19.
He's also found that everyone's network is laid out differently. Shown.
So if I put you in a machine, an MRI, functional MRI machine, and give you different tasks, I could tell you what's your age, what's your gender, how many languages you speak, whether you are right, left-handed, based on this representation. You don't have to crack the skull open to do that. But once you do, like in this case, like similar techniques. So this patient was trilingual. The mother tongue was Italian, and he later learned French and Spanish. And you can see that the representations are scattered here in the premotor area. Stimulating different regions can cause different losses in the different languages. So the bottom line is you have area specialization of functions in the cerebral cortex, and you can study this with all sorts of methods. And the MRI is a very powerful method to study the representation, and the concept of MRI is coming from the work of Roy and Sherrington. They noticed that the color of the surface of the brain was changing in different functional paradigms. So the active areas, they changed the color because of the deoxyhemoglobin proportions and also changes in the vasculature. So this is what we use now to localize functions in the brain. And if you put somebody into the machine and you hear words, then this language area is activated here in the temporal lobe. If you just see words, then different parts of the brain is active. If you speak words, then the motor areas are active, and you just generate words, but you don't vocalize, you don't produce sounds, then again, different brain areas active. So now we know where some of these functions are localized, although there is a distributed network of activities. And if you have a lesion in some brain areas, like we use this brain in Oxford for second-year medical students, you can see that this patient probably had some motoric aphasia because the left hemisphere Brodmann areas here responsible for that, Broca's areas, they were damaged. So the message is that cortical functional specializations are revealed with stimulation, lesion, and recording. So we know that these functions are localized in different parts of the brain. But how do we develop these maps, and are these maps permanent? And the answer is no. You can change the representation within the brain very easily. So, for instance, when you practice a language task, before and after this practice, you will change the representation and the activity pattern in the brain. Or there is a famous experiment by Jenkins when they were stimulating 3 fingers of a monkey 1 hour per day, and then they noticed that the representation in the cortex expanded corresponding to these 3 digits. Or you can do the same in human. If you do nothing else, just for 3 weeks, 10, 20 minutes per day, you stimulate some digits, then the representations will change also. And these changes persist in the brain. So I don't want you to think about the brain that, these maps, once they establish, they are permanent and fixed. They can actually, you move them around. But if you learn these tasks very early, then you have more considerable changes. So imagine that you are a professional musician. When you listen to music, piano tunes, and you are a pianist, then you even have motor cortex activation. You don't move your fingers. You just listen to piano music, but your motor cortex is active. Non-musicians wouldn't have this activation. Or you play on a soundless keyboard. You don't have sound, but very different cortical areas active for a musician and a non-musician. And probably you or your kid studied violin, and this is excruciating if you get it wrong, because you have to have all these feedback loops. So you have to listen to what you play, And you have to adjust your movements according to what you hear. And basically you build up very complex networks while you learn how to play the violin. And a trained and non-trained person will have very different activation in the motor areas when you listen to music. So my point is that early childhood training has big impact on your brain development. And the earlier you change it, the bigger the impact is. Let me show you some examples for this. Paško Rakic is one of our gods of our field of brain development, did this experiment about 50 years ago. And this is just from a review. So as I showed you before, the macaque occipital pole has a huge area dedicated for the visual perception, and that's the primary visual cortex. Now, if you remove the retina at embryonic stages from these primates, then the occipital pole will be very different. So a 3-year-old macaque with a normal visual cortex, flat surface, and this had no visual input to the occipital pole, you can see that the whole brain is very different. Now, you have to do these manipulations at early stages. If you do it, you don't have such change. If we cut sections from the occipital pole, you can see that the extent of the primary visual cortex much reduced. So the early experience has a big impact on brain development, and this impact is coming from the structure called thalamus. And this thalamus will interact with the brain during development, and that will change the circuit. So I don't want you to think about brain development as a computer board which was printed, and then once it's ready, you switch it on, and then you run your programs on it. The brain is not produced like that. The brain is changing according to the information you process. So, and also, your brain is switched on while it's developing. So, the activity you process in your brain will have a huge impact on the circuits you will have. And that's why I could tell a lot about some functional activities which we record, or even structural MRI from your brain. I could tell a lot about you because you shaped your brain during your life. And the experiments which came to demonstrate this came from Tom Boozy and Hendrik van der Loos. And you probably heard about the barrel cortex during your studies. So a huge area is dedicated for the representation of the whiskers of the upper lip. And if you stimulate the whisker, then you have activity in the corresponding region in the somatosensory cortex. And if you cut a tangential section through this area, then you will see these rings of cells in layer 4. And these rings correspond to the periphery. And let's hear it from Hendrik van der Loos directly. He's explaining how these were discovered.
Mouse to try to find out how and when the vital connections are formed.
Well, the mouse has 66 whiskers. There are 33 on each side of the upper lip. Normally the whiskers are in this situation. When the animal actively wants to use them, as we would use our hands when we go to an object, it moves them all in concert forward, like this. One could in fact compare these whiskers with as many blind man's sticks that a blind man would use, juggling them all to find his way in an extended tactile space. Now each of those whiskers is implanted in a heavily innervated sensor, a sensor connected to the brain.
Van der Loos discovered that each whisker on the face has a group of nerve cells in the cortex called the barrel that's exclusively devoted to that whisker's use.
In the microscope, one can actually see the correspondence between a pattern of whiskers on the animal's face and the pattern of barrels in the animal's brain.
On the face, the whiskers line up in neat, clearly distinct rows. In the brain, the same rows are evident in a slightly semicircular pattern.
Now I turn to a mouse from whose face a set of whiskers had been removed at birth. And what we see in this adult mouse brain is, in the place that corresponds to the whiskers removed, barrelless territory.
The missing barrels correspond precisely to the missing whiskers. Simply removing the whiskers changes the structure of the cortex.
When we take an animal between birth and 5 days of age and we make a very small lesion, the barrels that receive the signals from the whiskers that I have taken out, that these barrels are no longer there. So there is a profound upheaval in the brain. If we do the same experiment in an animal that is 6 days old, the brain will not respond at all. The map that is in the brain, the visible map, the barrel ensemble, will remain in place. So that means that the brain is now committed.
The whiskers of
So this concept was actually very powerful in the 70s because these experiments directly demonstrated that it's not the brain which had the information to generate these patterns, but you have to have a sensory input from the whiskers at certain stage of development to actually to shape the brain. So we subsequently understood that if you stimulate certain sets of whiskers and you inject 2-deoxyglucose to these animals or you image the activity, then certain regions will be corresponding to that activity. And we also understood how this pattern in the periphery is actually shaping the patterns in the central representation. And simply because these patterns are so apparent that you can see them with your naked eye on these flat mounts, this system became very powerful in showing how and when this activity is changing the pattern of the brain. So if you remove a row of whiskers, then you will have a corresponding change in the representation. But you have to do it in the first couple of postnatal days. You cannot do it later. Then the plasticity period is finished. You can also cut the infraorbital nerve, and then you no longer have barrels representing. And it took a while to understand that these interactions between the thalamus and the cortex shape the brain, and the thalamic input has to find the recipient cells and carve out these representations. And this has a particular time frame related to thalamocortical development. So first, the thalamic fibers are distributed in a homogeneous fashion. Then they get this clustering according to the periphery, in the signal in the periphery, and then they push the layer 4 cells out. So this is how you carve out the representation. So first the thalamic fibers and then the cells will move. And now we understand at what stages and how this has to occur to shape the brain. So after this introduction, now I turn to my work, what I have been doing in the thalamocortical circuit. So that's why we became interested in these very early critical stages, how the thalamic fibers interact with the brain. So as a student, I established that very early on, you have a very nice map of representation in the cortex. So if you trace with carbocyanine dyes from different parts of the brain, then you can backlabel corresponding thalamic cells. So the 3D volume is mapped onto the surface from the very early stages. Then we understood how the thalamic fibers arrive, how they accumulate, and then how they grow into the cerebral cortex at stages when the neurons are not even born, the majority of them. So we understood that this layer here, below the cortical plate, the so-called subcortical plate, has an important role. So the earliest thalamocortical projections shape your brain. So if we understand how the thalamic cells interact with the brain, then we understand these principles. So that's why I started studying the interactions between the thalamus and the cortex. And if you cut sections from the mouse or rat brain in this angle, then you can, in the same section, you will have the somatosensory thalamus, segment of the primary somatosensory cortex, and the interlinking pathways. So I use these sections to look at the evoked response using voltage-sensitive dyes. And if you stain your sections using these voltage-sensitive dyes, then you can reveal the activity pattern. So this is a newborn mouse brain. You can see the thalamus, the cortex, and the interlinking pathways. If you stimulate here, there in the thalamus, you can see that the evoked response in a newborn animal is localized in below the cortical plate, and it's a very long-lasting sustained depolarization. These squares, they flash with 10 microseconds. So you can imagine when I did these recordings 25 years ago, it was very big progress in the field to understand how the activity was interacting with the cortex. So you can actually image the spread of action potential in the tissue, and you can see the activity pattern within the brain. Then we discovered that this pattern is actually very time sensitive. So if you put a 2 days old or a 4 days old animal and you stain the brain slices and you line up the videos, you can see that the activity pattern is very different in a 2 days old than a 4 days old. In a 4 days old, if you stimulate the thalamus, you already have a very early response. It has a columnar arrangement and it moved into the cortex, whereas a 2 days old have a much longer lasting response, which lingers in the cortex for several hundreds of milliseconds. And it turned out that this activity pattern initially is targeting the subcortical plate, the subplate, and these are mediated through glutamatergic synapses because you can blow it away with a cocktail of glutamate receptor antagonists. So this is how we understood that this interaction with the subplate can be very interesting at the early stages. Later on, you also have huge changes in the first few postnatal days. Initially, you have this diffuse activity and then activity corresponding to the barrels. So if you want to change the pattern of the barrels, you have to do it at these early stages. You cannot do it later on. So this is corresponding to this plasticity period in the barrels. Now, it took about 20 years to understand what is really going on. This is the timing of the response, which is also changing. So the volume learning in the brain is changing with the age. And also the circuit is changing constantly. So initially, the thalamus is interacting with the subplate cells, and the subplate cells already feed some activity to the cortex. And then you have a transient circuit, which is coexistent with the adult circuit, which will be a thalamus layer 4 connectivity. But without this circuit, you could not establish this. And that's why I really became interested in these early subplate cells, because they have a very important role in forming these circuits. If you don't have these cells present, then you cannot finish the building work. So that's why pediatricians, they are so worried about early perinatal hypoxic ischemic brain damage, because these cells are damaged. They call it white matter damage, but it's actually the subplate cells are damaged, and if you have lesion here, then you cannot finish the building work. So if you delete these cells, or you silence them, or you manipulate them, then the barrels will not form. So here Patrick Cano did this with Kai Kaila in Helsinki. They used a complement-mediated ablation of subplate cells. They didn't touch the thalamus, they didn't touch the cortical plate, just the subplate, and the barrels couldn't get established in these animals. So what are these subplate cells? So subplate cells have been described by Paško Rakic and Ivica Kostovic, and then later Marla Laskin and Carla Schatz did some pioneering work on them. So these subplate cells are the first generated cells of our brain, and if you birth date them, and you have a look early after birth, as Marla and Carla did, then you see a large number of these cells. But if you wait a few more weeks or months, then these cells were disappeared. Only a few interstitial white matter cells will remain. So there, if you do a similar birth dating for the adjacent layer 6 cells, then they actually remain. They don't disappear. So there's a preferential cell death in this cell group. That's why if you study the adult brain, you never come across with them, or you don't study them, or you don't look at them. But I became obsessed with these cells during development, and also understanding their adult function, because you do have a few cells remaining. The only difference is that in primate, these subplate cells, they remain as interstitial white matter cells, whereas in the mouse, you have a compact cell layer called layer 6B. But the gene expression, connectivity, and other parameters are very similar between the two cell groups, so they are homologous to one another. And we had a look at many other species, and I just show a few which we did with Paul Menger in Johannesburg. And in all of these species, you have a certain proportions of interstitial white matter cells, and it's very similar to humans. And there is area variation in the number of these cells. So why are these cells important? Now, for this, we have to move to pathology. If you have a look at the interstitial white matter cells in schizophrenia patients, then in the prefrontal cortex, you have a much larger proportion of these cells surviving. And although these studies are quite early and they use very relatively primitive markers for these neurons, but the result is clear. You have a much larger number and different distribution of these cells in the prefrontal cortex. So the remnants of the subplate cells are very different in these pathologies. If you look at the autism patients also, you have a much larger number of the interstitial white matter cells, and also their distribution is different. So these cells are the shadows of the subplate. And that's why I gave this title to my talk, because I'm studying these cells. How do they cause these pathologies? So they are not just developmental remnants of a developmental population of cells from the subplate. They have some very important roles in the adult cortex. And the last 15, 20 years, that's what I have been studying. What is the role of these cells in the normal cortex? So if you have a normal development, you have certain number of cells surviving. If you have a reduced activity or asynchronized activity due to all sorts of pathologies, then you have more cells remaining. So how do they cause the pathology? And for this, we had to use all sorts of models, including animal models, to target these cells. And to understand their function, I have to introduce the concept of the first order, higher order thalamus. I don't know whether it's taught in medical school, or in Oxford nobody knows usually the difference, but you will immediately understand why it is essential to know this concept to understand the function of these layer 6B cells. So the thalamus have relay cells, and they are mostly the first order thalamic nuclei, like lateral geniculate nucleus, medial geniculate nucleus, or ventrobasal complex. So the sensory signals, they come through these thalamic nuclei, and they reach the primary cortical sensory areas. And you also have a huge volume of the thalamus, especially in primate, which is not getting direct sensory input. Most of the input is coming from the cortex, and they relay it back to other cortical areas. So there is a transthalamic cortico-cortical communication via these higher order thalamic nuclei. Pulvinar is a higher order thalamic nucleus, and you remember from the dissection room, pulvinar is huge in human. So what we discovered was that layer 6B has a very special relationship with the higher order thalamic nuclei. So layer 6 is projecting to both first order and higher order thalamic nuclei, and layer 5 is only going to the higher order thalamic nuclei, and we discovered that layer 6B is very similar to 5. They go to the higher order thalamic nuclei. Now, what is the relevance of all this? Most of the time, you don't listen to the environment, you don't care what's going on around you. Very occasionally, you update it with the sensory input, but most of the time, you have you live in a kind of a dream world. You think that you understand what is going around you, and mostly you listen to your own brain. So 90% of the input to the thalamus is coming from the cortex, and only 10 is from the environment. But occasionally you have to kind of reconcile stimuli because you don't quite understand what's going on. And then you want to activate more cortical areas to resolve what is the unusual situation. And then you have to wake up this system. And I would like to convince you in the next 10-15 minutes that this is actually coming from the layer 6B cells. So they are the ones which help you to wake up the higher order thalamus to have these more complex cognitive functions. And if you have too many of these cells or too active, then you become locked into this system, and then you start have illusions, delusions. And if you have too little of them, then you don't activate this, and then you don't process information when you need to do that. So we discovered that the layer 6B cells, if you cross them to a reporter line, so in this particular Cre expressing mouse line, most of these cells are in the bottom of the brain, but you have some also scattered into layer 6. And their projections are exclusively going to the higher order thalamus. You see they leave the lateral geniculate nucleus and the VB empty. They go to the pulvinar and the PO here. These are not cells, these are just their projections. You don't have cells in the thalamus. So this was one discovery that we have now a third type of pathway which is going to the higher order thalamus, different from the adjacent layer 6. They have very extended connections in the brain. We used Cre-dependent adeno-associated viral tracing from these cells, and we showed that they have a huge network. They don't have side branch to the thalamic reticular nucleus, which is an inhibitory cell population. And also, we looked at the size of these boutons, and they are much smaller than the layer 5, which is also projecting to the higher order thalamus. We don't know what is the relevance of this, that they are smaller, and they don't have this side branch to the thalamic reticular nucleus. So they are very well suited to wake up the thalamus. Now, who is talking to these cells? For this, we used monosynaptic rabies tracing. As you know, the rabies virus is jumping through synapses. That's why it's so deadly. But we used modified rabies, which was developed by a group in San Diego. I don't remember the name, right? And basically, we just used the same technique. And you can see that you use these cells as starter cells. And then once you express the receptor, then you inject the virus, and then the virus will jump one synapse. So if you use the layer 6B cells as starter cells, you can see who is talking to them. So we use this method, and when we injected prefrontal cortex and we used these cells as starter cells, we noticed that Arthur's favorite structure, the hypothalamus, was lighting up. So basically the hypothalamus is directly regulating these layer 6B cells when to switch on and off, which was, for me, it was a huge surprise. Of course, mostly layer 5 cells talk to them, but you had some input from the hypothalamus. And these cells were the orexin expressing cells. As you know, the lateral hypothalamus have a few thousand cells which produce this peptide orexin, and they stabilize the arousal. And what is interesting is that the only cell group in your cerebral cortex, this huge structure, is the layer 6B, which is responding to orexin. You have some interneurons in the cortex, but it's only the 6B, the projection neurons. So, and also the higher order thalamic nucleus have more orexin sensitivity than the others. So as you know, if you don't have orexin, or you don't have the receptor for orexin, then you can have cataplexy. So you have these videos on YouTube where a dog is becoming very excited, but they don't have orexin, then they have cataplexy, and they fall asleep immediately. So that's why layer 6B is such an interesting population, because orexin is acting on them. So could orexin act on this network, this cortico-thalamo-cortical network, which is waking up your brain to process outside stimuli? And that's for this. We decided to silence this cell group, genetically manipulate them so that they cannot signal the thalamus. So how do you do that? How do you silence layer 6B neurons? So for this, we used SNAP25 conditional knockouts. As you know, in the synapse, you have a snare complex, and this complex is essential to release the vesicles in a regulated manner when you have an action potential, so when the calcium goes into the synapse. And so we decided to remove one member of this snare complex, the molecule SNAP25, which was discovered by Michael Wilson. He died, he didn't get the Nobel, but he could have because of this discovery. Basically, without SNAP25, you cannot release the vesicles in a regulated fashion. And so with Michael, we did a conditional knockout, and we also knocked out SNAP25 in these different layers in the brain, including layer 6B. And we studied the behavior of the animals. And if you knock out SNAP25, you have a very normal initial development, but depending on which cells you silence, some of these cells are actually dying at quite late stages, but not 6B. They stay alive, they develop normally, but they are silenced. So what is the symptom of these animals? So today I'm only talking to you about the layer 6B silence, but we studied all the others, and we discovered some interesting sleep changes. But if you silence layer 6B, then you reduce anxiety of these animals. So in all sorts of paradigms, they behave very differently. So if you silence the input to the higher order thalamic nuclei from layer 6B, then these animals have no change in circadian rhythm, or when you put them into the dark or you give them jet lag, they behave similarly. But when you look at their sleep, the theta and sigma ranges are abolished. They sleep otherwise normal length of time, unlike layer 5 when you have reduced sleep. And if you look at the behavior of these animals, they avoid normally the open arms of these elevated plasmase, but not the layer 6B silenced animals. They still go out to these without any problems. They also go out to the light side of the light-dark box, and they also have a reduced novelty preference. So if you put them in a Y maze and you only expose one arm, then normally a normal animal, when you open this arm, they would go here preferentially 80% of the time. Not the DRD1A Cree silenced, layer 6B silenced animals. They go to the same arm 50-50, so they have a different behavior. Orexins and anxiety have been known, but it hasn't been known that it might be the layer 6B which is mediating this anxiety behavior. So we are very interested in the possibility to target this particular interaction, orexin and 6B, because then without manipulating this system, we could actually change the behavior. And that's what we have been studying over the last 5 years now, how anxiety is mediated. And if you record from brain slices where you silence layer 6B, then orexin cannot act on these circuits. If you put your brain slices on these multi-electrode systems, so every blue dot here is an electrode, and you pour orexin, then these brain regions in the prefrontal cortex are active. If you silence the 6B cell populations, then you don't have such activity. And we believe that this anxiety behavior is mediated through these cell groups. So what we are doing now is we are looking at all these in human sections, and we showed that there is a similar behavior of these cells in the interstitial white matter cells, and this highly exposed image showing that there are no cells in the amygdala which express this Cree. So we are not touching the amygdala this anxiety behavior is mediated through the cortex. We are also doing in vivo recordings from 6B cells in various behavioral paradigms, and we want to see when they are active. And we want to examine this possibility that how we communicate with the environment is actually mediated through these cells. So somebody with more or less interstitial white matter cells, like in autism and schizophrenia, has a very different percept of the environment, how they perceive novel objects, or how do they process sensory information. And we believe that maybe some of these altered development of the brain has an impact on these circuits, and layer 6B is important in that. So if you have a developmental abnormality, you regulate the cell numbers and these circuits differently, and then this will have an impact on the rest of your life. So we also study models where we have more layer 6B cells surviving, and the last 6 years I have been visiting professor at Charité in Berlin. Einstein Foundation was funding this position. And basically with Matthew Larkham and Britta Eichhorn and Tim Zolnik, we have been looking at a model where we keep more subplate cells alive. It's a P10 conditional knockout. We have 30% more 6B cells, and we are looking at the behavior of these animals. So this is an autism model, and we study that. So I hope I convinced you that your own brain is kind of unique, and it's shaped by your conditions during development. And the very early stages of your brain development are associated to these transient cells. And depending on how you regulate the number of these cells during these very early stages of in utero and early postnatal development, you can actually have altered life. So these early circuit adjustments can have a very important role in your later life. And that's why they are so important to study. And that's why we study them. The developing brain is not just a smaller version of the adult brain. It's a very different structure, time frame, plasticity, critical periods. So the first 1,000 days of your life have a much bigger impact on the rest of your life than the other 1,000 days. So that's why we study them. So I'm very grateful to my department and founders and collaborators over the years. And if you are interested in this topic, I recommend this book to you, which we wrote together with Joy Hirsch and Tamás Horváth. It's not selling very well, so hope you buy some of some of these. Thanks for your attention.
Well, thank you so much, dear professor. So I don't know what can I say now. So your lecture has taken my breath away. So in this way, so absolutely fantastic, awesome piece of neuroscience. So any questions from the audience? No? So it's a mistake. It's a mistake.
Don't scratch.
Hello, thank you for the lecture. I really enjoyed it. It was very nice. I have a question. What would happen if the brain plasticity remained on the same baseline level as it is like in the beginning of the 2 days, and it would remain on the same level throughout the whole life? What would happen then?
Fantastic question, and we probably don't know the full scale answer, because as I showed you, lots of things change during development. You remember the video I showed you from the 2 days old or 4 days old mouse? You stimulate the thalamus the same way, but you could see the pattern is very different. So this is a hypothetical experiment because we can't do this, because cells come and go, you have transient structures, myelination kicks in, interneurons mature. I didn't even talk about microglia, astrocytes. They also change. They also have sex specificity. So basically, I think it would be a chaos, no? If you retain plasticity. My brain would be very slow, no? And I would start associating with all sorts of things with everything, no? Because you could see that a 4-day-old animal, already the response was half the time than a 2-day-old. And probably it would be chaos, no? We wouldn't remember anything. We couldn't retain memory the same way. So, yeah, it's a very interesting question, but I think there's too many things to consider.
Do you think it's possible to like reactivate the plasticity again?
To a certain extent, yes. You could reactivate it with all sorts of manner. Even just some of the microtubules, if you reactivate that behavior for spines, that would change. If you inject astrocytes, that can reactivate plasticity. Microglia can have an effect on all these. Most of our subplate cells die, so our circuits after the death of these cells is very different from when they are present. So if you want to reactivate ocular dominance columns, with or without subplate, you have to use different strategy. You know these experiments when Carla Schatz ablated subplate cells and showed that ocular dominance did not develop. So these cells are responsible for some of these early patterning. But when you want to re-establish plasticity in an adult, the subplate is already gone. So that plasticity will be very different from the plasticity you had when these structures were established. So that's why I'm humming here, because it's very difficult to imagine a situation when all these cells come back for the plasticity. So we have to find a way to alter plasticity without these scaffold, transient dynamic scaffold cells around. And that's why it's so difficult to treat cerebral palsy, because if you have a hypoxic ischemic brain damage, perinatal stages, then you remove these cells before you finish the building work, and then you establish this condition which is unfolding over the subsequent years, spasticity, and it's very difficult then to rearrange the function so that you don't have the symptoms. So that's why it's so difficult to treat cerebral palsy in later stages of life. Yeah, I think you touched on a very important issue.
Yeah, thank you very much.
Thank you.
So and maybe just my question. So this involvement of orexin signaling seems to be especially intriguing in this context. So do you know many other neuropeptides may interact in this process, in this development of this connection?
So I must say that hypothalamus was not my favorite structure during medical school.
I understand.
But my respect grew enormously, especially with the interaction with Tamás, who is a hypothalamic expert. He discovered some of the mechanisms related to...
Tamás Horváth, I know him.
Yeah. So we are actually from the very same city. And we went to the same nursery, same primary school, secondary school, but he's 2 years younger than me, and he was the best friend of my younger brother. You know, when you are 6 years old and you have a 4 years old, you don't deal with them because they are just chipmunks. So it's really later when we became good friends with Tamás, and he studied ghrelin, leptin, and other structures and these circuits in the hypothalamus. And I didn't pay much attention to these structures, but when I started looking at the receptor distribution of these hypothalamic peptides, and that's your question, in the circuits, I was shocked that these hypothalamic mechanisms act directly on circuits which are involved in your highest cognitive functions. You know, not basic sensory functions, but if you look at the distribution of orexin receptors or leptin receptors or neurotensin or even dopamine, it is very specific cell groups which are under the influence of these neuropeptides from the hypothalamus. So during evolution, I think we produced a huge structure, the cerebral cortex, but it's still very much under the control of the hypothalamus. And also in your T-shirt, you have the
Psilocybin.
Psilocybin. Psilocybin is acting directly on layer 6B.
Oh,
And
that's
the other cell group where psilocybin has the action is layer 5. So it's not a coincidence why this is so selective. So you can imagine a situation that you activate these layer 6B cells with some of these cannabinoid receptors, and basically you have a situation when you activate these cortico-thalamo-cortical systems, and this is how you get this travel during this situation. So I'm amazed that some of these neuropeptides which are released from the hypothalamus, and I always thought that they have a non-specific activation of the brain, they actually know exactly where they are going and which nodes of these circuits they stimulate. So although our brain is, our cerebral cortex is 80% of the volume of the brain, this 0.3% volume of the hypothalamus has a huge impact on the rest of the structure. So the short answer to your question, all these peptides, they have very specific effect, and 6B is very unique because of the orexin, neurotensin, dopamine activity.
Yes, so thank you so much.
Thank you.
Thank you, Professor, for the lecture, and thank you that finally you point my lovely glial cells. So this is my question. Is there any research, or maybe you're planning to check the cooperation between the subplate cells and the glial cells during development, for example, the radial cells?
Superb question. Subplate is in a position to alter myelination because it's very close to these fibers in the intermediate zone and also later in the white matter. Subplate is neurosecretory. If you look at the single cell transcriptome of subplate cells in many different species, you have all sorts of molecules which are released from subplate, including connective tissue growth factor. And CTGF has an effect on myelination, and I don't think anybody studied this in detail. They had a look at the myelination of the CTGF knockout of the whole brain, where there was a moderate change in myelination, but not tragic. But I think if we could selectively alter layer 6B cells and the CTGF they produce, we would get some kind of a alteration in myelination. Myelination comes in at the time when most of the subplate cells disappeared. So there might be some relationship between the number of subplate cells and subplate activity and the regulation of myelination, and nobody really looked at this in great detail. We had a look at the myelination and also the astrocytes, oligodendrocytes, and microglia in the models where we silence these cells using the conditional SNAP25, and there are huge changes in oligodendrocyte behavior, division, and what is also interesting is depending on which cell type you silence, you have bigger effect on myelination. But the biggest effect for us was coming from layer 5 and not 6B, I have to tell you. So if you silence layer 5 cells, the myelination is changing more than 6B, which I expected originally. So, and also we looked at the microglia, how they respond to the silencing. And it's interesting that, again, there is cell type specificity. If you silence layer 5, the microglia goes in, the density goes up in that particular layer, so they notice that these cells are silenced, and then they start chewing up these cells and their synapses because they are silenced. So there is some kind of signaling going on between the silenced cells and the microglia. And what was very surprising for me, and I think for everybody, it's sex specific. If you silence a layer 5 cell, in a male or female mouse, the microglia will have different responses. I couldn't believe this. Auguste Vadishiute did this, who came from Vilnius to my lab and did a PhD and now doing a junior research fellow position. And she discovered it based on just a handful of animals, that there is a sex specificity in this response. So I think you are absolutely right. It's not just because we manipulate neurons, this will have a huge effect on all the other cell types of the brain as well. Thank you.
Okay, that's all for this topic. Thank you so much, dear Professor.
Thank you.
Again, it's amazing. Natomiast wszystkim przypominam, że tydzień mózgu wciąż trwa. So the brain